Our nervous system is made of different types of nerve cells. Some are involved in the process of thinking, memory, while others are responsible for muscle contraction and movement. These are called motor neurons, as can be seen in B, and can be compared to a control center that will send the command to move, and the muscle will execute the voluntary action. The resulting movement is therefore completely dependent on the quality of the communication between the motor neuron and the muscle. So can you imagine what can cause the loss of this valuable communication? Well, this is exactly what leaves people with amyotrophic lateral sclerosis. Patient will gradually paralyze, having difficulties performing movement as simple as walking, talking, and even eventually breathing, condemning them to die within 2 to 5 years once diagnosed, as shown in A. During my PhD, I just lingered to analyze the communication interface between the motor neuron and the muscle, called the neuromuscular junction, which is illustrated in C. But it is important to know that a third entity, the glial cells in green, is also part of this dynamic interface and act as a mediator. So I wonder whether glial cells can properly fulfill their role as a mediator in this ménage à trois in the context of the disease. Because we know that in a healthy person, glial cells will be able to hear and to adjust the level of communication that takes place between the motor neuron and the muscle in order to harmonize their dialogue and promote better execution of movement. In a situation of injury, where the communication is reduced or even interrupted, glial cells will detect this change in the communication, will restore the dialogue, and will promote the recovery of movement. My results have allowed me to discover that in the case of amyotrophic lateral sclerosis, glial cells are the equivalent of deaf because the receptors that serve as their ear are defective. So how can a mediator, deprived of his listening skill, can understand, promote, or restore dialogue? Hence, this dysfunction of glial cells has a direct impact on the quality or the very existence of the communication. My research significance is then of the greatest importance since my results have identified a novel therapeutic target in the disease. I even hope that one day my result can have a significant impact on the quality of life of patients. The idea here would be to restore auditory abilities of glial cells, restoring their function as a mediator, and help patients to move and live longer. Thank you.